So we will be going through what is that? Mm, I guess you're able to see the presentation.
No, sir.
Yes, no, what is it? Yeah, okay,
Yes, sir.
okay, okay, thank you. Yeah, we will be going through this, the genetic molecule, that is the genetic molecule. We're talking about nucleic acids, one of the important one. Okay, as we had discussed earlier, the blueprint part that is there, which is having all the information, describing the characteristic, describing the feature of an organism, feature of an of the particular animal that is there, be it the organism, bacteria, microorganism, any space, any part. Okay. Now, whenever we are making any kind of molecule, one of the ability it should be that it should be passed down from one organism, from one generation to the next. Okay, then it should be easy to make a copy. All right, so the quality should be there whenever we are talking about any kind of genetic material. Genes over here is like basically characteristics of an organism, feature of an organism, trait of an organism that is there. So when we are talking about a trait, and if that trait is represented by a molecule, that molecule should have some stability. That means it should not be changed while either while transmitting it from one organism to the next, or while a copy is being made of it. Okay, it should be remain exact. So like for example, you know the game Chinese Whisper or something, wherein 10 people are in line, the first person says something, the second person has to repeat that, third and fourth, so on, and the last person has to again repeat it and tell out loud what exactly was the message. And during that transition, if there is any change in it, automatically the message changes. May have seen some memes or videos or comic video on that. Now the point is about genetic molecule. When it is being passed from the first person to the last person, it does not change. It remains the same. Okay, nothing more, nothing less, as it is. That is what a molecule will be considered as. Okay, you are the genetic molecule. Okay, that is the reason the blueprint. The blueprint is something like a master copy. So has everything about the particular design we are talking about. So in this, we will go through certain parts again. So in this, we'll be talking about DNA as we had discussed last time, RNA, okay, the full form that is there, deoxyribonucleic acid. RNA is a ribonucleic acid. They are macro because they are very, very large molecule. DNA, when we take it up, comes approximately 2.2 meter long. Okay, approximately. So then the smaller molecules from which DNA, RNA molecules are made. Now, what our last time we had discussed that what they are made of. They are made of your nucleotides. One unit of a DNA, one unit of an RNA is your nucleotide. Okay. Then DNA, RNA are the different type of nucleic acids. So since many such nucleotide molecules are joined together, they are called as polynucleotide. Okay, they are called as polynucleotide. So in this, three things are there. Okay, the prime one nucleotide will have your pentose sugar, 5 carbon sugar. Then the nitrogen base, nitrogen containing base, a big molecule. which will have this. Then a phosphate group is there. Okay, then that is how it is. This is how it is attached, as we had discussed earlier, I think. So over here will be your oxygen. Over here will be the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Okay, so five carbons, that is their position. So this phosphate is connected with your fifth carbon. Nitrogen is connected to the first carbon. Okay, that this is a nucleotide. Anyone remembers what is a nucleoside? When I change this to S, what will be missing from it? Or what will be added in it? किसी को याद है क्या? Now, अगर मैं इस चीज को निकाल दूं, if this is removed, if the phosphate is removed, what is left is the sugar and this base. Then this becomes your nucleoside. So I guess one of you answered correctly. The rest all are sleeping ahead. So next part. So what is happening? The phosphate and sugar form the backbone of the rung. Now we'll see what a rung is, what a step is. So the point is that this that is there, these form the backbone.
So your voice is lacking.
Are not able to hear? Thomas, are you able to hear now? Or Chinmay, who else was that? Am I audible now? Yeah, is the voice clear?
Yes, sir.
All clear? Yeah, I think it was Chinmay or Thomas. Who was it who raised that? Or is it voice lag? All right. So as I was saying, the backbone, meaning the main structure, the one holding the whole thing together, that is between that bondage what is there between the phosphate and sugar help. Okay. So yeah, we'll see. Let's move ahead. So basically what is happening is the DNA, as we talked about last time also, it is a double helical structure. So it has two strands together. So this is one strand, this is another strand. Okay, one, two. So these form the rung of what? Rungs of a ladder. If I make a ladder like this is the step. This is what is called as a rung. See, a ladder. So there's a green thing that is there, that is your step. The blue thing that is there is your rung. Okay. So this is how a DNA helix looks like. You take a rope ladder. Now over here, when we talk about like a rope ladder that is there, and when we twist it, we get this output. So this structure is called as a double helix. Helix because of this helical fold, how it is folded. Double because there is two strands are being bound to each other and twisted in it. All right. Now we will see each of these parts later on. So over here, what we see is that this pairing, this bonding between two nitrogen base. So there are four nitrogen bases we had seen last time. There is A, G, T, C. So this bonding between the two is called as a base pair or BP. Okay. So if a question comes like if there is a strand, there is a DNA sequence with DNA with a length of say 100 base pair, 100 BP, 100 base pair, then that means how many nucleo nucleotides are there? Can anyone give a calculation in the chat? How many nucleotide? Who all are it? I hope everyone got the question, what I'm asking. Did anyone understand what I'm talking about or forgot what we discussed last time? Okay, this is one base pair. Now each of these is bound to a nucleotide, okay, attached to it. So if one base pair includes two nitrogen bases, then 100 base pair is equal to how many nucleotides? I see two of you are replying, Khushi and Devangi. Where are the rest? No more response. Okay. Shruti, okay. Khushi, Shruti, Devangi, you three are right. Good. Okay, earlier slide we saw that one nucleotide, Prathamesh, correct. One nucleotide contains one nitrogen base attached. One base pair is between two nucleotides. One base pair is equal to two nucleotides. Why? Because one nitrogen base, one nucleotide, two nitrogen bases that is forming one base pair, two nucleotide. So 100, when we say there is 100 base pair, that means 100 into 2 is equal to 200 nucleotide. So those who all wrote 200 in the chat are correct. All right, good. So this attachment that is there between the two strands is called as base pairing. Okay. Now base pairs we got it. Now the type so nitrogen bases that are attached, adenine, this is one. Then there is cytosine, thymine, and guanine. As had explained, this one and this one is a purine. This one and this one is a pyrimidine. Okay. So one, as we had discussed earlier also, the rule that whenever there is a binding, Adenine always binds with thymine. Cytosine always binds with guanine. Okay, one other specific base. This is specific only for DNA, what we are talking right now, the blueprint, the genetic material. Okay, so adenine will bind with thymine and cytosine with guanine, forming base pair. Each of these two that is there. Okay, now coming to the structure of the DNA. As we saw earlier, they saw that this is made up of two strands to each other. So over here is your sugar. That is sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And another one with a sugar phosphate. So these are opposite. That is your base pairing is that G will always bind with C, A with T, T with A, C with G, then like that A with T, T with A, like that. So that is what it is. So these are the bases. These are the bases. Now this sequence that is there, this sequence represents G. We have got this sugar and phosphate as the same. It is a four type of nitrogen bases. Their sequence, how they are arranged. Okay, that is in this case, what is the sequence following GAACAT? Okay, GAACAT. That is how it is. So this represents a gene sequence. So that is what the blueprint is. Once we get to know the sequence, we can figure out what it will form. We can figure out what it will make. So that way we can monitor it and regulate it. Okay. So what this statement is saying, they are complementary. That means the order of the sequence of bases in one strand determines the other. So you pick either this left one or the right one. Okay, there is left one or right one. So the sequence of left one will determine the right one. Sequence of right one will determine the left one. Okay, that is what complementary means. So if you take one of the strand sequence, if you know the sequence of one of the strand पता है तो अग दूसरा strand को automatically पता चल जाएगा. We will be able to figure it out. How? The base pairing that is there. C pairs with G, T pairs with A pairs with T, and G pairs with C, or C pairs with G. Okay. Now, in the chat, give me the sequence of what will be the alternate sequence for this particular strand that has been given. What will come over here? Just write the whole sequence and give it to me. I got from Kushi. Don't give an individual chat, individual text and give for the whole sequence. Just write the sequence of the nitrogen base present on this side. Okay, Kushi, Shreya, Devangi. Quite good. Dipendra Yadav. Shravani. All right, let's see. Quite good, quite good. Wow, Niranjan, not bad. So, okay. T gag TCA. That's what I'm getting everywhere. T gag TCA. Let's see. There's a T gag. This A will always be with T. C will always be with G. T will always be with A. C will be with G. A will be with T. C. A. So we got T GAT TCA. Okay, right? Or ACT GAT. Anyway, मैं जो so whoever those who all gave है Shruti, Niranjan, Prathamesh, Josna, Khushi, Shravani. Good, very good. I'm glad that you guys are getting it, understanding it. Yay, it is correct. Nice. Now, can someone tell me what is the error in this slide? Or there is no error in this slide? Wow. You are like Khushi ma'am, you are typing and shouting with caps. Okay. They should be anti-parallel. I understood what you mean. Others? Prathame, phosphate direction of second, opposite. So what you are writing, phosphate direction of second, opposite. Phosphate should be down, so downwards, anti. Ah, so if I am correct, what you are saying, so from that, this should have been something like this, what I drew. If you agree, please give a yes. I'm just sad that I had to bring up the question. Nobody said anything. Anyway, so good that you all understood when I asked you if there is an error. So yes, this part that is there, this should be facing in the opposite direction, ulta. Okay, reversed. As you guys said, anti-parallel. So you guys remember. Very happy. Boom. Attached. There goes our DNA has been made. Okay. Now, genetic code, I think I told you about what codon is. Codon represents three things that are there. All right, three letters. So that is that. We need to look at a sequence of bases. That is what we're talking about. So triplets that is there. There's three sequences, three letters. That sequence of these three nitrogen bases that are there represents a represents a particular this codon that is there. It represents an amino acid. Okay. This amino acid is represent. So then this sequence, if I say there is, I'm just spitballing here. Okay, this is not the exact one. So this is the sequence of DNA. All right, we need to convert it to mRNA. From mRNA, we will get it to tRNA like that. Oh, we'll come to that again. So codon. Codon is what which that represents your amino acid, which will then help in forming the protein chain, a polypeptide. So that is what codon is. What the sequence that is opposite to the codon, the sequence that is opposite to the codon or complementary to the codon, not opposite, complementary to codon, will be called as anticodon. Okay, complementary. So if this is codon, then this will be anticodon, complementary, AGGTCC. So this is complementary to the codon sequence. Okay, so so again, coming to this part, the base pair, the sugar backbone that is there, this is how it looks. All the arrangement that is there. Okay, of the DNA structure forming. So DNA is what is there. One of this strand will be used for making a wire. Now, two strands are there. The two strands are folded in such a manner to form a double helix, and the helical structure is formed. By this adenine to thymine and guanine to cytosine. That is the sequence, that is the pattern. All right. And there is no particular order in the sequence that After adenine thymine, it has to be guanine cytosine or cytosine guanine. It can be any order, it can be anything, but the pairing is always between thymine, adenine together and guanine cytosine. Now, in this case, between adenine thymine, there is double hydrogen bond. Between guanine cytosine, it is triple hydrogen bond. Okay. Now let's check out what they have to say. Wait, are you guys not able to? Are you able to hear the sound now?
Yes, yes.
Deoxyribonucleic acid, or DNA, is a biological molecule that contains the genetic code used by all known living organisms. Similar to a recipe or a blueprint, DNA contains instructions living things need to develop and function. Four different molecules called nucleotides are the units of DNA. Because of their structures, A can only bind to T, and C only binds to G. DNA is structured as a double helix of two complementary strands of nucleotides. The two strands can be pulled apart, like a zipper. And because each base can only pair with its complement, all the information of the double-stranded sequence can be obtained from a single strand. DNA is the code of life, but it also contains historical
Aman ka
information
number
because as generation after generation reproduces, it collects mistakes called mutations. By comparing DNA sequences between different living things, researchers can determine their relationships in the evolutionary tree of life. And not only is DNA a blueprint, its structure is important too. It is a building block researchers can use to design nanostructures that, given the way DNA works, will basically build themselves. Scientists are also creating computers that use these qualities of DNA to solve complex problems in a new and interesting way.
Okay, so about DNA's importance, how it is, sequences and all. If you have seen movie like Jurassic World, Jurassic Park, DNA has been utilized a lot in a technology called as genetic engineering, biotechnology. Okay, all right. So that's what the mutation is like. If I change what the video was trying to say was, if I change a sequence in this part, the whole sequence changes. Like if it is say A T C C G A T T A T. All right. Now this sequence represents, say for example, eye color. Eye color for brown. Now what I do is somehow I change this C to G. Because of this change in one sequence, now this 3 that is there will represent a different amino acid. Since the amino acid is changing, different amino acid has come over, so the protein structure will change. As a result, maybe instead of brown, the whole eye color changes to, for example. So you see how DNA plays an important role and what all applications there are there of the DNA that we will come later on in the biotech. Now coming to the other aspects of the DNA structure. All right. Now the DNA that we know, DNA that we are studying, or the most commonly found type of DNA in the organisms and all, is generally called as bDNA. Right. Now in this bDNA means B type is there. If B type is there, that means definitely there would be something called as C, D, A, whatever. Okay. Now in this type, what has been shown over here, there in this image, there are certain numbers. What are they? Okay. Now you see there is like this is base pair. This represents one base pair, BP. That is what we use, base pair. Okay. It's like you can say kilobytes per second or like that, BPS, what we use for the digital world. So this one base pair, there is another base pair over here. Now the gap between the two base pair, right? This gap is called as rise. Okay, is called as rise. How much is that? See over here. This 0.34, 0.34 nanometer. It can also be represented as Armstrong. Okay, I multiply it by another 10, and then I get Armstrong, A and a degree sign. Able to comprehend what I am discussing here, guys? Just give a yes or no in the chat, please. Yeah, all right. See, earlier I was discussing the general structure. What all parts are involved in it? Okay. Now what I'm talking about is in this structure, how do we figure out what is the length? How did I know that it is nearly 2.2 meter or something like that? On further study on microscopic studies of this using crystallography, spectrometer, and other devices, we found that the gap has been approximately is zero point 0.3 nanometer. Okay, and this is one base pair. So rise is what? Rise is, you can say, the distance between two base pairs in a DNA. That is your rise. Okay. Now these strands that are there appear parallel to you. Okay, they appear parallel, right? But on closer inspection, it is found that they are not exactly parallel. They might be like this, they might be like this, or they might be like this. So what I've drawn here, this, that means what? Can I say that these two, if this what I've drawn here represents a base pair, all right? Again, this represents a base pair. Okay, this represents a base pair. This strand, this line that is there, 1, 2, 3, 4, 5, 6, like lines are there, they represent. Now, can I say that if these two meet, they will make an angle? If you all understand till here, please say, type in yes. Yeah, okay. Yeah, they make an angle, right? Now, if they meet or not, that means there is a chance they will intersect. So that is an angle. So this angle is called as tilt. Tilt is kind of the angle made by the two base pairs, two adjacent base pairs, not any two adjacent. Rise is what? Distance between two adjacent base pairs. समझ रहा है ना? चलो बढ़िया. Okay. अभी now okay. So now we saw what is the gap between the two, what angle they are making, all that. Now what is there? What now the turn that is there? Yeah, they can be parallel too. Okay. Now You are able to see these words written there: major groove, minor groove. All right. What is this exactly? Is now they are making a helical turn. When I say this, okay, make a straight line. Two strands are there. They can be like this, right? Equal. No. What? Rather, it is not like that. It is twisted in a particular fashion, right? It is going this way, and the another complementary strands goes kind of parallel with it to a certain, and then twists. So when it is twisting, you can see there is a big gap over here. Big gap over here, and a small gap on the opposite side. So this big gap is called as major groove, and this small gap is called as minor groove. This again helps with attachment of the proteins to the DNA, these grooving patterns that are there. One then again helps in folding. Okay, this is how it is. So this is a straight chain. That is what a helical structure. So major groove, minor groove. Now in within a turn, you will find now this is one complete turn. This what I have drawn, complete turn. So one complete turn will have a major and a minor groove in it. Yeah. Okay, Saloni, yeah. Groove over here is a gap, right? Gap thingy being produced over here, V shape, and this is like a small V shape. All right. So this like a groove. That is what groove means. Basically, is gap only. Similar thing, but then we call it as a groove so that it becomes an anchor for attachment of, you can say, the protein to a certain, yeah, proteins and all other structures binding to it. Okay. Also, it folds in such fashion for stability. Okay, there is a phosphate, there is a sugar and all. It folds in this pattern for stability. Now coming to the next part, क्या था? हाँ, ये जो है, what I was talking about, this line and this line. So if I count, let me see how many are there. 1, 2, 3, 4, over here 1, and over here 1. 10. Okay, like 1, 2. So in this one turn, the DNA that we have in our cell, one turn, this is called as a one turn or pitch. This is called as a one turn or pitch. समझ रहा है? So it's like you take a rope ladder and twist it. So it will follow a similar twist. So in this right diagram, what I've done, this is one turn, this is one, like that. So here, this when it is twisting, it's twisting in a different fashion. So it will have one major, one minor, one turn or pitch. Okay. Now, what is the length of this? In this DNA, one turn consists of 10 base pair. Okay. One turn has 10 base pair. And the distance between the two base pair, as I told you, is 0.34 nanometer. So can you make a guess what will be the length of one turn if each turn is having 10 base pair? Yeah, anyway, the answer is given over here. This is what the answer is. So it's ridiculous, like idiotic to ask you also. It's like a rhetorical question I'm asking because there is already an answer given. Yes, 3.4 nanometer. This, though, that means one turn's length is 3.4 nanometer, or this is Armstrong. Okay. Yeah. So this is the unit, and you can get questions on this also a lot. All right. Say our DNA sequence is there, which has got 2000 turns. If 2000 turns are there, calculate how many sugar molecule is there. I just made up this question myself. Prathamesh, what have you written? 6800 nanometer for? And Khushi ma'am, you have written 40,000. Okay. Shreya ma'am says 40,000. Niranjan says 20,000. Hey, Niranjan sir, the question is about sugar molecule. Nobody is asking about base pair. Now, if this question has been changed to how many major groove is there, then wow, majority of you got it correct. Niranjan, now you are correct. Ananya sahi hai. Devangi sahi hai. Khushi ma'am is right. Shreya ma'am's right. Yeah. If I say 200,000 turns, then how many major groove? Okay, Khushi ma'am, you are saying 2000. Devangi ma'am is asking me, is it 1000? I am asking for an answer with a full stop, not a question mark. Shreya ma'am says 2000. Pratham says 2000. Niranjan 1000. आई 1000 कैसे हो गया 2000 टर्न है तो फिर आधा कैसे हो गया one turn has one major groove, one minor groove. So if I say 2000 turn, then it will be 2000 major groove and 2000 minor groove. So how are you getting 1000? Yeah, yeah, 2000. Yeah, if the question has been changed now, instead of major, just asking how many groups, then how many, then what will be the answer? Antara ma'am, nice. Saloni ji is nice. Kushi ma'am, as always, बढ़िया. Yep. Shruti ma'am, how did you get 8th? No, Shruti ma'am. See, okay, I've made a mess over here in the diagram. Let me just clean this. Let me just clear everything. Okay, see, this is one turn. One turn. One turn has two grooves. In this two, one major. Yes, Prathamesh, I will tell you that. One major plus one minor. Okay, so if there are 2000 turns, so 2000, so there is 2000 major, 2000 minor. So एक turn में दो है तो 2 into 2 हो जाएगा 4000. Now 2000 turns, one turn has 10 base pair, correct? One base pair is equivalent to 2 nucleotides. One nucleotide molecule has one sugar molecule. So can I say one base pair is equivalent to 2 sugar molecule? Question is 2000 turn. One turn, the 10 base pair, correct? One turn, 10 base pair. That means 2000 turns into 10, that is 20k, correct? One base pair, two sugar. That means 20k into 2 is equal to 40k. Questions, and I'm not saying it will come always with 2000 like that, but this correlation between the turns, correlation between the grooves, correlation between the base pair number can be asked. All right, in such kind of numerical can be given, numerical type questions can be asked in your NEET exams. Okay, so these values are expected from you to remember them. The question may not provide it. Okay, that one turn will have this much and all, and based on that. Okay, now coming to okay, just a minute. Okay, can you guys see this? Okay, you guys are able to see this. All right, now this is about type of DNA. This is the type of DNA that I was discussing. All right, the type of helix, right-handed means what? It is folding on the right side. Left-handed means opposite side, obviously. So there are two other types of active DNA structure that has been observed. Okay, that has been observed. Now, this is the most commonly found DNA in organism. You want me to zoom? How do I zoom now? Oh, okay, got it. It's my bad. This is fine. Yeah. All right. So as I was explaining about this, the helical diameter means when we observe from the top. Okay. That is what this helical pattern, the folding, like you have studied something called as right hand thumb rule, right? So the folding that is going in that direction, that's what. So that is what right-handed helix is, left-handed helix is. So when I am having this fold, so what is the, if I observe from a top, now over here I'm giving a side view. If I observe from the top, as you can see over here, wait, why am I unable to draw? I can just show you this. You can see this part over here. This is the helical diameter, which is coming to 2.37 nanometer. All right. Rise per base. Rise means, as I told you earlier, the distance between the two bases that are there. This in bDNA is 0.34, in aDNA 0.29. Now over here, the diameter of A is little less, whereas in the rise per base in A is also little less. Whereas zDNA it is highest. This is another one. That is the 3.37. More the rise means more base pairs will be there in the turn. That is what rise is about. That is what the distance. Okay, distance per complete turn. Okay, complete turn, as in one complete turn or pitch that is there, completely twist, one complete twist that is there. The distance or the length you can say is 3.4 nanometer here, 3.2, and because there is more rise in the basis, so it is 4.5. All right. Now, A and Z, we will see the design or approximate diagram and illustration in the below image. All right. To show it, see, this is how it looks. B, A is kind of more constricted or condensed. Z is nearly appearing opposite of what B is. Now, mind you, these two patterns are observed, all right, among organisms in certain circumstances. A, like for example, has been observed in, like, what is A is considered as if like more condensed form of B. B is the standard form that we are considering. All right, B is the standard form that we are studying. B is the standard form that we are observing. B is the standard form that we take into consideration whenever we are discussing any aspect or any concept with related to the DNA, like DNA replication process, okay, or say protein synthesis process that is there. Hmm. So this is what. Now, what has been observed is when B is more condensed, loses water and becomes more condensed or constricted, results in formation of aDNA. Okay, like for example, some organism has been pushed to a very extreme condition in certain region, certain habitat, like some bacteria, for example, has been found to be in extreme dry condition or something. So there is desiccation, loss of water. So to protect it, the DNA, the bDNA transforms or converts to be a DNA type. Okay. Okay. Number of base per turn, 10 base pairs that is there. 10 in B, in A it is 11, in Z it is 12. Now, topology meaning over here, how it appears. Now, I showed you the diagram of B earlier, right? The major groove looked like a very wide V, and whereas the minor groove looked very narrow V. Now, deep or shallow that is there. Okay. Uh, Wait. Now, topology is what I had shown earlier. This is like quite wide and deep, going deep towards the folding, whereas this one is narrow, not that wide, and not either deeper into the structure. That is what topology term is trying to explain. Okay, that is a topology. Then comes a minor groove, major groove. In this, it is narrow, deep, broad. It is flat. Like we don't see much of a folding pattern in that, or much of a grooveness in that. Okay. Now coming to this, I hope you guys have taken a screenshot of this. Anyone here who didn't get the idea of what A and Z DNA are? Okay, and ZDNA has been observed to be formed generally during transcription process. Okay, in certain cases, it has been found in the brain section in Alzheimer disease. You know, so during transcription, what happens? The DNA is unfolding, and while refolding, it takes the reverse twist. So over here, in general, you can say that if B is the main structure, what we're talking, A is like a constricted form or more of the bDNA. Z is more of the opposite form of the bDNA. You can see the twist is, for example, it is right-handed, this is left-handed. Okay. All right. So got it, all of you. I will then move to the next part. Okay, guys, this was about types of DNA and all. Then coming to the next, RNA. RNA, basically the same thing. Two difference comes in RNA. One is the sugar. Okay, I hope you all are okay with the DNA thing. All right, the structure, the parts that are there, the grooves that are there. Okay. Now, RNA, coming back to RNA again, as we had discussed last time. One is uracil is present instead of thymine. Okay. Secondly, this that is there, this one is not missing the oxygen. It is the sugar molecule is having the oxygen. Two major difference. Another one is another difference can be added as that RNA are generally single stranded. Okay, single stranded. Like, okay, if we talk about this, see over here. So you can see this. This is RNA. This is DNA. DNA is like making a helix. Two strands are joined together. RNA, whereas, is single stranded, making a fold. All right. And instead of thymine, there is uracil. Okay. Second difference, what I said is in this part. Who is that? Where is that? Is the diagram here? Okay. You can take a snap of this, guys. All right. It gives like a structural difference between the RNA and DNA. And I think Khushi ma'am, you asked about a second difference. What I said was difference in this molecule. See, this is ribose. This is okay. So that is what deoxy. That means one oxygen is missing in the ribose sugar that involved in making DNA. Another difference you can say here, this is single stranded. You can see one, and this is two. Literal two strands are present in DNA. In RNA, what happens is generally it is single stranded. There can be binding, that is base pairing can occur within itself on folding. Okay, like I had shown you last time, the tRNA. And in some rare cases, like in the plant viruses that are there, RNA acts as a genetic material. That time it can be found as double stranded also, but rare. In the case of eukaryotes and all, like us, that is there, it is, well, RNA is single stranded, DNA is double stranded, DNA is the genetic material. So going back to this RNA, so the three functional types that is there is your messenger RNA, transfer RNA, ribosomal RNA. mRNA, as I had explained earlier, generally it is made in the synthesized within the nucleus from DNA by a process called as transcription. Previous slide. Okay, ma'am. Done. Okay. So okay, so yeah, mRNA that is there joins up, helps in transcription. It is generally linear. This is single stranded. Doesn't undergo infolding, like folding among itself either. All right, and it is two people in the waiting room. All are in. Okay. Hmm. So this transcribe that is there is your transcription. Okay. Ribosomal RNA, cytoplasm with ribosome, hence ribosomal. It's an RNA, but generally bound with your ribosome that is there. So your ribosomal RNA helps with the translation process, the second step of the protein synthesis. This also we had discussed last time. tRNA, Job, we had discussed the structure of tRNA, the hairpin structure, and the clover leaf structure, the uh, arms of the tRNA, what they are doing. What is the job of the tRNA is to bring the amino acid to the ribosome, that is during the translation. Now tRNA will contain the three nitrogen bases which are complementary to the sequence on mRNA. All right, and it will have the sites where amino acids are attached. So this tRNA is bringing a new amino acid, then this chain is added, and then so that is how the protein synthesis is there. And the protein synthesis is a different part, but then yeah, what is the job of tRNA? Helping with amino acid recognition, helping with joining of amino acid forming polypeptide. Yeah, Prathamesh. Yes, they have anticodons. So over here in this, what do you see this UGG? If I take this is codon. So this will be your anticodon because UGG, what is complementary to it? ACC. So it is anti means like it's not means like it is a complementary part. That is how it is. We are giving the terminology. So this will be your anticodon. So this will represent the sequence or that is there. So there's triple A, then GAU, then UUC. Then coming the next, the next, the next. Okay. Well, this is self-explanatory, so you can just make a note of it or take a screenshot of this difference between the RNA and the DNA. The important aspects to remember regarding, see from here you can even note the properties of DNA. First of all, it is always double helix, right? This sugar is deoxyribose, or one oxygen atom is missing, right? The nitrogen bases are adenine, guanine, thymine, cytosine. This thymine that is there is the one key factor over here. Now, this is very important. Ratio of adenine to thymine and the ratio of cytosine to guanine will always be 1. Another thing you can do is A plus G is always equal to T plus C. Okay. This law that the number of purines and number of pyrimidines in the DNA is same equal was given by a guy called as Chargaff, and this is called as Chargaff's rule. Based on this, you can get a question too. Like, like nuclear, what is there are anyway, we'll make a question later on. Right now, I'm getting a question point. So, point is, this is what the rule is. It will be the same. So, that is what this statement also is directing towards it. That whenever we take the count of adenine thymine, it is always 1, and that means they will nullify because the number of adenine and thymine will be same, number of cytosine and guanine will be same. And since G and C is same, hence A plus G and T plus C is also same. Okay. And another part about this is that RNA is chemically less stable and it is very stable. Now, this is where the problem comes if The RNA is the genetic material. Since RNA is less stable as a molecule, there is high rate of mutation. Now, if RNA is a genetic material, like for example, like virus, the coronavirus that we have. So, since it is made up of RNA, the genetic material, and it is unstable, so it becomes difficult to make a vaccine because of the changes. The mutation can occur, resulting in making different kind of protein coat, and as a result, well, difficult to fight it off. Okay. In this, it is ratio is not same. It can be the number of adenine uracil can be vary. Number of adenine is different. Uracil is different since there is no base pairing. Another part is it is ribose. Oxygen is present in this pair in this pentose sugar. Another aspect is there is uracil and there is no thymine. So A, G, and C is same in both, but uracil is different. Phosphate compound is same. Okay. I guess quite self-explanatory. Rest all. So you can see the difference where the difference is in the oxygen. It is in the second carbon hydroxyl group is present in ribose, whereas in the deoxyribose, hydroxyl group is replaced by H. And this difference, this is a very minute difference, right? I'm just exchanging hydrogen with hydroxyl. I'm replacing this hydroxyl group. This change in this thing, in this one single change, drastically alters the molecular property or the chemical property that is there. The deoxyribose becomes much more stable compared to ribose because of absence of this hydroxyl group. All right, and hence DNA is considered more another property comes in that it is more stable. One is a double-stranded molecule, so all the free nitrogen bases are bound. One, then the sugar molecule is also stable. So as a result, the reactivity or the reaction is less, and it stays kind of, you can say, inert. All right. ATP, well, had explained earlier also, adenosine that is there combines phosphate. When one phosphate is added, it is well, ATP is your adenosine monophosphate. Another phosphate, 2 diphosphate, and 3 that is triphosphate. This is the energy molecule. Anyway, this again we will discuss. It will come across in the chapter respiration and photosynthesis. So when we are joining these, we are getting a polynucleotides. Now over here that is there, the difference is in the second carbon OH. Previous slide, which one? This one? Okay. So the these two are involved. So I think we have discussed this. So now I'll go ahead. So phosphodiester bond formation, that is between the two sugar molecules. So in the left diagram, what we see is there is this fifth carbon binding with this phosphate, and this phosphate that is there is binding with the third and the fifth, and both these connections are ester. And since there is two ester linkage occurring with the phosphate, so you can call, hence it is called as phosphodiester. What happened? Can you speak up? Clear? Ligase enzymes are those groups which act like Fevicol, joining things. Okay. So 5 prime, 3 prime. I guess you guys remember this. So basically we are trying to figure out what does the word 5 prime that is there, 3 prime mean. Wait, let me put this on. Please write in chat if you're able to hear it.
Hi everyone. A couple of people have asked me on my biology forum by email in the last couple of days, what does it mean when we're talking about DNA? What does it mean when we talk about the 3' end and the 5' end of DNA? This is something that obviously is a bit confusing to people. And so I wanted to just explain that fairly briefly. I think it really helps if we start off by looking at a single nucleotide of DNA. And we're going to look at not only the 3' end and the 5' end, but the structure of a nucleotide. And we'll look at what it means to talk about the 3' and 5' end in that context. There's 3 parts to every nucleotide. Firstly, you can see that there's this, most importantly, this 5-carbon sugar molecule, in this case deoxyribose. We'll come back to that in just a moment. There's also this phosphate molecule with the yellow phosphorus and the oxygen, which is red. And then in addition to that, there's a nitrogen containing, but the silver bits in this particular model that we're looking at here are representing carbon, and the little white bits are hydrogen, but they're probably less important. So the nitrogen containing base, the phosphorus in the phosphate, and then down here in the middle, we've got our 5-carbon sugar. Now, if we look at that 5-carbon sugar, and we start numbering the carbons from where they join the base, okay, so let's do that. I'm going to write this on here. So here's our nitrogen-containing base with all the blue nitrogen in it. That would make this carbon number 1. This one here is carbon number 2. That would make this one carbon 3. This one's carbon 4. Sticking off to the side here at the top is carbon number 5. Now you'll notice that the phosphate is attached to carbon number 5, and that gives us our 5' end of this nucleotide. The 3' end is where this hydroxyl group is, this oxygen with a hydrogen on it. That's our 3' end because it's attached to the 3rd carbon. Does that make sense? Now if we look at a polymer of these nucleotides, because remember DNA is a long chain of nucleotides, a polymer of nucleotides. So if we look at this, you can see that obviously this would be the 5' end of that nucleotide. Then this here is the 3' end. That phosphate there is attached to the 5' end. There's their 3' end, and then sticking down the bottom here is the 3' end, which gives us a 3' end of that whole chain. So we would say that this whole chain of nucleotides, this strand of DNA, this is the 5' end of it, and that's the 3' end of it. Just going back before we leave this, while we have such a nice diagram of a nucleotide on here, I do also just want to point out to you, when we're looking at the bases here, that remember, we can tell which base this is by looking at a couple of things. Firstly, there's a couple of places where hydrogen bonds here can form. Two of them, because there are two hydrogen bonds that can form, that would mean that this is either adenine or thymine. If it was cytosine or guanine, there would be three places where hydrogen bonds can form. Okay, so how do we tell whether it's adenine or thymine? Well, you remember that some of the bases have two rings in them like this one does. You can see there's one ring there and one ring here. And the ones with two rings like that we call purines. The ones that have a single ring we call pyrimidines. And you might remember that the bases C, U, and T are the pyrimidines. This one, of course, is adenine, so it must be a purine. Okay? See ya.
All right, this clears the part regarding it. All right, so enzymes we have discussed last time also, so I can see that pretty much so many of you have an idea of what enzyme is, how it is working. So we will just brief through the enzyme part. So now enzymes, depending on enzymes functions can be within the cell, though it is made within the cell, its function can be within or outside also. If it is working within the cell, if it is, what is it called? Yeah. If it is working within the cell, then intra. If it is outside, extra. That means it is released out. That is what it is. Released out of the body. Anyway, now proceeding. That is what intracellular, extracellular enzymes are for. Now, lock and key hypothesis, as we'll just briefly proceed on through as well. All right. Now, enzymes that are there, they are made of your proteins, and also they have a certain structure. Generally, all enzymes are globular rather than fibrous. They are like folded within. So when they fold in, there are like that grooves are formed or structures are formed in which the substrate comes and binds. So that is your active site to which the substrate is coming and binding to it. Within that, as binding and catalytic sites are there, right? So generally the hydrophilic groups are on the outside of the molecule, so they're soluble, have a special feature that is active site to which the substrate comes and binds to it. Okay. Now proteins, as the diagram was shown earlier also, enzyme and substrate forming an ECS, that is enzyme substrate complex. And from this, once it binds, product forms. And since the product that is there is different than enzyme, different substrate, it is released out of the enzyme. So in this case, what happens is the substrate forms a key which fits the lock of the enzyme. The substrate is held in place by the bonding of the amino acids that are forming the structure, the groove within the enzyme, all right, due to the functional groups. And this hold together, that is the enzyme and the substrate together, form what is called as the ECS or the enzyme substrate complex. So enzyme generally is specific. All right, each type of enzyme will usually act on only one type of substrate molecule and is said to be specific for that particular, is considered to be specific for that particular substrate. That means that this enzyme will have this substrate. It will not have any other. All right, there was a lock and key model which was discussed, and then the model lock and key was too rigid, but it was found that they are showing some flexibility. So another model called as induced fit hypothesis was there. It's just the difference is that in induced fit, when the substrate is coming, the enzyme is slightly modifying to accommodate the substrate. All right. So over here, that is there, the substrate binds to the active site. Now, active site over here, there is a conical structure, pointy apex is there, but the substrate is not having that pointy part, neither on the other end. So when the substrate is attached to it, the enzyme residue, the amino acid parts that are there, they reshuffle or readjust to accommodate that part of the substrate. Now over here, this that is there, this section and this section is not same. So substrate when it enters or comes to attach to the active site, the apex part of the enzyme is accommodating it. So this is what induced fit is. When the product forms, okay, a question by Kushi. Not accurate, it is accurate. What it is trying to say is that lock and key is what? In the difference, lock and key is totally exactly the same thing. Okay. That means in lock and key, if this shape is like this, the substrate will also be like this. So, but then the thing is in what has been said is that the substrate enzymes are not that rigid. Lock and key is like exact fit. If even if there is a little bit of 19 it will not adjust. But induced fit model describes that there is little bit of fluidity. Okay, little bit of what is it called? Like you guys remember the fluid mosaic model that was explained for cell membrane? How it was showing little bit of adjustment to allow the movement of the proteins within the membranes so that you know it and the stretchability is there in the membrane due to the lipid bilayer and all so that it can accommodate somewhere. Okay, so induced fit says that there is little bit of adjustability is there so that it can fit in. It's not exactly to the substrate. That is what it says. And lock and key says it is exact, like Funnius be saying. Both the models are accepted. Induced fit is more accepted nowadays due to being that giving that minute range of adjustment in it. So both are right. Both models are accepted. Because of the specificity of the enzyme, the lock and key is also accepted. Okay. Uh, now, okay. So here it is. Sequence of step catalyzed reaction, enzyme binding to the substrate, forming the enzyme substrate complex. And when the reaction is done, the product forms, product is released. So again, enzyme is free to react again. So this is the whole cycle. So because of this, the enzyme reusability is there. So it's beneficial. Many fast reactions can be done and repeated reactions can be done. And substrate can be interchanged. So over here is an example which actually gives an idea of how enzyme shows little bit of flexibility, or you can say adjustability in this case with the enzyme lysosome. So enzyme is there, substrate is binding to it. So is an enzyme defense against that polysaccharide chain that forms a cell wall. Okay, in this hydrolysis reaction. So it is binding to it. All right, it may not be exactly as it is. The groove it is sliding in it, but the groove is showing some little bit of adjustability. That's what it's showing. And then the product is formed, it is broken down, and again a new, that is the new carbohydrate can be brought in as a substrate and broken down. Right, this is an example of how an enzyme is working in hydrolytic activity. Now please answer this in the chat. Is it A, B, or C? C. Okay, Aditya says A. Aditya ji, read the question. Okay, majority of you are going for C. Some are saying they are protein. I guess you're not reading the question correctly. It's a very easy question. Which statement is correct for all catalyst? It is talking about catalyst. Catalyst के लिए कौन सा सही है? हर एक कोई catalyst enzyme होना जरूरी है क्या? They work in हर कोई catalyst living organism में होना भी जरूरी नहीं है. Yeah. Yes, somebody raised a hand. Kalyani. Yes, who was it? Raised a hand.
So by mistakenly that.
अरे कोई नहीं, ठीक है, तो पूछ भी सकती है. Okay, guys, I'll see the answer. It is C. See, catalyst, as soon as we hear the name, it is increasing the rate of reaction. That is what the idea we know. Okay, there are catalysts which are non-enzymes too. All enzymes are considered as catalysts. So all enzymes are catalysts, but all catalysts are not enzymes.
So, but catalysts change the rate of reaction. Na? It is not necessary that always they will speed up the reaction.
Yes, okay. एक और चीज MCQ solve कर रहा है. If you're solving MCQs of the four lot, which is the most correct one? See, this doesn't fit. That all catalysts. The question is on. Yeah. And yes, catalyst by definition, they change the rate of reaction is a general one, and there are catalysts which are set to reduce the speed of reaction. Yes, but they are called as negative catalyst or inhibitors. But generally, whenever we use the word catalyst, now like catalyst, if it say somebody saying he is a catalyst for something, or like we are using as a figure of speech or adding as a metaphor, a simile like that, it's always considered like somebody is increasing the speed. Somewhere it is just like going up, not down, not slowing down. Okay, that is what's what. And what you said, who was it by the way? Who said that? What you said is correct that it is altering the speed. But now we have to check the question, what it is saying. In the question, what we get is it is asking for all. That means it is in general. Are they all enzyme? Not true. Are they all protein? Not true. Are they all living in working in living organism? Not true either. So of the four, which is the most apt? C is the most apt. So when you're solving an MCQ question, multiple choice, always don't go by that. Okay, but definition says this, then I cannot pick that up. You have to pick the choice which is the most apt one in that particular question of the lot that is given to you. All right, guys, that is one of the rules you should follow for MCQ solving. I hope it makes sense. Okay. This one is pretty easy. Enzyme is 3, substrate is 1. Yep. All of you got, like whoever typed in got it correct. Josna, Koshi, Dipendra, Niranjan, Shravani. Cool. Super. This one. Okay. Quite apt. Very easy. Good. So you're getting the idea. Now coming to this point about how enzyme is working on how this catalytic activity it is doing. So basically what it is doing is it is reducing the activation energy. I think I have to explain earlier also that whatever energy required, the minimum energy required for a reaction to be proceed ahead, all right, for changing the reactant to product, that we can say is as the activation energy. Now enzyme is completely reducing that. So it's basically, it's like you on a road, on a flat road, there is a car present, which is needs to be pushed, and we need 5 people to push it. All right. Suddenly, what has happened? This road has been made slopey. So automatically, you don't need 5 people. 1 or 2 people is enough to just start the push or something. Okay. So that is what cat. So that the energy required, or the people required, or the work part, that is reduced. Okay. That is what it is about. So what this graph shows is that the reactants are present at a level, they have certain energy, and then the energy level is increased. So this is your activation energy at the tip of this bell curve that is there. All right. Okay. So this is the part going up and then going down. This is the general progress of the reaction. And enzyme will do what? Will just cut short this activation energy that is required for the reactant to reach the point and then change to product. So over here is what energy is released. So it is like an exothermic reaction. Hence the product's energy level is lower than the reactant. Yeah, Devangi ma'am, what's your question? Third point. Your previous slide or this slide?
Previous slide.
In this, ma'am, Benedict's test is something, a test done for, what do you say, detecting the presence or absence of reducing sugar within the given sample. So when we heat it, the temperature is there. It is the reactants that are there in it. They are getting the energy to react, and as a result, color change occurs. That is, if we just leave it. So basically, what in this case it is there, you need to heat the reagents so that they can react with it. That's what it's saying. So basically, what is it saying is that how are you giving the activation energy? One way is to provide heat. Another classic example is a cooking, right? We are giving heat to it. We just by keeping the cut vegetables together, they don't get prepared, right? We are giving heat. So heat is like we are giving energy, which is raising, providing some sort of energy, which will then raise them to the excited level, and then they will react. As a reaction, whatever nutrients are there, they will react and give out the aroma, and the food will be cooked. Similarly, in this case, a chemical reaction that is occurring in Benedict's reagent. All right. So basically, what the idea is of the activation energy is somewhere either we are providing heat, or we are providing, or the activation energy is provided. Generally, we provide heat, is provided by some form. So enzyme is reducing that. Okay. In the case of biology, it will be like ATP. ATP is required. So say for example, without the enzyme, it needs such high level of ATP to give the energy. When enzyme is there, the ATP count drastically goes down. You can see the difference between the two. It's quite, well, high. It's at this level, and here it's like almost less than half. So here we don't need to provide that much of heat. Here we are providing heat to the substrate so that it heats, gets prepared, and then we are getting the product. So this much quantity of excess energy is required to obtain this. What enzyme is doing is it is reducing that. The interaction, how it can be done, the interaction with the amino acids present within the enzyme helps in that point in reducing the activation energy. So as an example given, now there is an incline over here. That means what energy is required to climb that short incline, then reach the bottom. What is the an enzyme doing? It is just removing that incline, the hump over there. Done. You just give a slight push and then boom, it goes down. Okay, that is what it is doing. Effect of catalyst. Same thing over here. So the enzyme is getting speed up because it is lowering the activation energy. So the time is saved, energy is saved, and whatever this excess energy is there can again be utilized in doing further enzymatic actions. So this, if you go to this, that is there, holding the substrate. It's the groove of the active site, you can say. The how the amino acids are arranged and how the enzymes, the polypeptide is folded. The functional groups or amino acids are exposed to interact with the substrate. That is where the point comes. That's what gives the enzyme the ability to decrease the activation energy, because the amino acids that are there, they are in such a way that already their reactive groups are exposed. So as soon as substrate comes, they are ready to react. So hence you can say in certain way the activation energy is reduced. Now try to solve this. I would suggest please read the questions and the statements properly. It is asking which is wrong, so let's see. Only two of you have answered, rest of you confused. Shreya ma'am says. All right. So Shruti, खुशी, प्रथमेश सही पकड़े, शमिका, श्रेया गलत पकड़े हैं. Okay, let's see. This is what is it asking? False. What is false? Which is incorrect. Enzymes are protein. Yes, they are protein. They function as catalyst. Yes, they do. Yes, Kalyani ma'am. कुछ है या वापस गलती से?
गलती से
ठीक. Okay, so they are specific. As we discussed earlier, they are specific for a substrate. So that is again correct. Enzymes provide activation energy. Are they giving activation energy? No, they are lowering activation energy. They are not giving energy, nor they are raising the energy. They are lowering it. And enzyme is not providing any kind of energy source. It is not providing any kind of. So third statement is wrong. Let's check out the fourth one. Enzyme activity can be regulated. Enzyme activity in the sense their rate of process can be reduced, increased. Yes, that can be done. So last statement is again true. So false statement is C. So your खुशी, श्रुति, प्रथमेश, बस बाकी सब गलत, ये तीनों सही है. Okay, next, relationship between an enzyme and a reactant can be best described as. What? You can't see question? All can see question, right? Yeah.
Yes, sir.
Yeah. Okay. Already started. Kushi, Devangi, Shreya, Shruti, Shravani. Hmm. Okay, let's see. Yep. All of you got it correct. Good. So so relationship is temporary association between the reactant over here is your substrate. So the substrate enzyme association, the relationship is temporary. All right. It is not stable. Yeah, I just showed it now. Okay, lag again, the lag. Next, same thing. Reactant and the substrate both are same. Okay, effect of concentration, enzyme concentration. This is again the graph is same, same in the sense it goes up, similar fashion that as the enzyme is increasing, reaction is increasing. So it forms a proportion into straight line. Basically, this is the graph it will make. So till here, enzyme will be the limiting factor controlling the reaction. After a point, if since the substrate, excess substrate, if substrate molecules that are there are no more, that means that whatever substrate was added, we are keeping the substrate concentration same. So as substrate is there and enzymes are more, but then no more substrate to change, so it will go flat. Okay, this is about enzyme concentration. Oh, Kushi ma'am, your query with this. Okay, what I will do is okay. So back to this about react about the enzyme, how enzyme is affecting the rate of reaction. Okay, since we add more enzyme, more active sites, but then more high enzyme is there, their active site is free because all substrate are booked, whatever substrate is there. So hence it will reach a flat line over here, saturation. So at this point, sorry, at this point we call this as substrate as the limiting factor, because now whatever enzyme can do to increase the speed has done. आगे substrate enzyme और limit नहीं कर रहा है. Enzyme is not restricting the speed anymore or the velocity of the reaction anymore. Substrate limit has reached, hence this part is substrate. So since this, another thing is that generally in the graph this is called as the first order of reaction, this section of the graph that is there, because over here now enzyme is the one controlling the rate of reaction. More the enzyme, more the, after a while what happened, become mixed. Enzyme alone is not controlling the reaction, there is another factor jumping in. So this can be called as second order of reaction. Okay, sorry. Now next part that is there, effect of substrate. Now enzyme we saw more the there is a linear progression that is directly proportional. More the enzyme, more the reaction. Less the enzyme, less the reaction. Same way for substrate concentration, the effect is what? More the substrate, more the rate of reaction. As we are similar effect, as we are increasing the substrate, the rate of reaction is increasing. Vmax is the maximum velocity at which that particular enzyme can work. So in this case, what is there? Less substrate is there, enzyme is there. So as we are adding the substrate, reaction is going on. Same over here. Here, this point, the middle graph shows there is saturation. Okay, it has reached that pinnacle point where the number of enzymes saturate and the number of enzymes and substrate is same. Now, even if we add more substrate later on, the blue color substrate, there are no more free enzyme to bind or react to it. Okay, so hence then we get a flat line over here. So till here, who is the one controlling the or regulating the rate of reaction? Substrate. Because as a lot of enzymes are there, but as we are adding the substrate, rate is increasing because free enzymes are there to bind to the substrate. Here, what is happening? Both are at the same part. Equilibrium kind of, you can say, has been reached. Here, no more increase. Even if we are adding more substrate, but no increase. So then, who is the limiting factor over here? Substrate alone is not the limiting factor. Substrate is no more the limiting factor, but the enzyme becomes a limiting factor. We need to increase the enzyme concentration again to counter the excess substrate concentration and keep the increase the rate of reaction. Okay, similar fashion as the enzyme. Okay. Okay, this question not required. Yeah, temperature and enzyme activity. Now, how temperature is affecting the enzyme activity? In this, we'll straight away go for the graph. It all is there. No. Okay, right. So, how is temperature affecting? Well, it makes a bell-shaped curve, as we had discussed it last time also, that as the rise in temperature, it affects the kinetic energy. More the kinetic energy, more the energy is there. So it will help in increasing the rate of reaction. But if the temperature is too high, it will lead to denaturation or breakdown of the enzyme itself. So it will stop. So hence there will be a drop later on. Now, raising the temperature one way helps in increasing the rate of reaction, but beyond a point, what happens is the molecules that are there, the covalent bonds and the other binding bonds that are there within the enzyme, they start rupturing. They start breaking apart. So when they start breaking apart, the whole shape of the enzyme starts shifting. So since their shape is shifting, they will not be able to bind to the substrate anymore, or the reverse, substrate will not be able to attach to the enzyme anymore. So as a result, enzyme becomes inactive and the reaction goes down. Okay. So the temperature at which the enzyme shows maximum efficiency or maximum rate, that will be called as optimum temperature. This is an important part. Now, our body temperature is kept at 37, which is lower than optimum, just to give that window. By chance, the temperature rises, the body is able to handle. That's the reason when our body temperature goes beyond, like, say, if we talk about Fahrenheit, 98.6 is there, goes 9900 and all, we are able to still survive because enzymes are working. If we always keep it at optimum and slight up or down goes above that, enzyme will not work at all and we will just immediately may die or will die. Okay. The optimum temperature will vary for the enzyme to enzyme, but there are organisms which have a higher optimum temperature. Like certain bacteria can, the enzyme will not denature even at say 70 degree, 90 degrees centigrade. So it can withstand that. Okay. One of those enzymes is a Taq polymerase, which is used in PCR test. You may have heard of PCR test with respect to the current pandemic. That is a COVID test. Yeah, what changes occur is that body temperature is rising because of the activity, respiration is going on. That's the reason that statement is mentioned over here, that it is kept at below optimum, or close to optimum. Because if a body temperature rises, automatically it will affect the enzymatic activity. So body temperature rises, metabolism is increasing, and our energy count or calorie count, we are burning more. Reason being, we are reaching towards the optimum part. So activity is more when the body temperature is higher, but then too high temperature is bad for us. Will just denature or stop the enzyme from working. Okay, try this out. Option C. Shreya ma'am, yep, chances hai. Where we are filled with all so much of lipids and all that. Yeah, spontaneous humus combustion can occur due to it. Can be triggered. So now the answer C, A. Niranjan and Shravani taking A. The question never mentioned when it had started, just said increase by 10 degrees. What if it was 0 and then 10? It might increase. What if it was at optimum and then 10? So I think apt answer should be C. It may increase or decrease because they have not mentioned any other information that whether it was at optimum, it was not at optimum, where it was. So in this case, increasing by 10 degree, it may affect positively, may affect negatively. So majority of those who all gave C answer, quite good. Shreya, Devangi, Shruti, Ananya. ओके इसका क्या होगा? श्रेया जी सही पकड़े, खुशी जी भी सही पकड़े. Ananya, I need ABCD. But anyway, you're right. Okay. Because we are talking about temperatures, obviously the temperature. अरे सलोनी मैम, काहे क्यों? कैसे pH को हम लोग नहीं बदलेंगे? We have to keep. We cannot keep the temperature constant. Okay. See, the test tubes are there. Yeah. Exactly. Thank you, Shreya. Exactly. In the question, it's mentioned that we are investigating temperature. If the one we are investigating and we keep it constant, how will we know what is happening? So, yeah. So, what you need to do is read the question, all right? And, guys, one tip, the questions are coming in English. So, read through certain books on English, and because there are word plays are there too in certain questions, like in the options and all. So, you need to understand that too. So, we need to work on improving our English as well. Science point of view, scientific English that is there. Okay. So if you can take help of this, find a book called as Word Power Made Easy. That can help you out. Okay. Okay. So yeah. So in this scenario, the example given of ethylene glycol, it is that yes, inhibitors are of use, and in this scenario, it is saying that drinking ethanol is good in a way, saving life, and inhibitors are required. Unnecessary reaction will not take place. For example, you studied about, okay, say for example, pepsin. Okay, now pepsin that is there breaks protein. Okay, pepsin is an enzyme that breaks protein. If it is always active in the stomach, then what will happen is it will eat up the protein layer within the stomach and may damage the stomach itself. Another example, say blood clotting. Okay, I'm not sure you've studied blood clotting in your 10th. Yeah, there is a chapter, right? Life processes, RBC, yeah, circulatory system. Yeah. So in that blood clotting that is there, there is formation of fibrин from fibrinogen. If fibrinogen is always getting activated to fibrин, then in our body there will be clot forming here and there anytime. And due to that clot, it may block the passage of the blood in the artery and vein, and we may die. So we need somebody to inhibit them, keep them inactive. So inhibitors are useful. It's not just for the sake of it. Oh, why, like that. Okay. Hence, that inactive form of enzyme is there, deactivated form of enzyme is there. They are lying there, but then so that there is no accidental problems coming in if they are always kept active. Okay. So yes, their use is there. So next, non-competitive. Non-competitive means they will not be competing with the substrate for the same binding site. Well, that is the idea. Okay, but they will bind to somewhere else and create a problem. Now what they will do is they will distort the structure, or they will alter the structure by binding to the enzyme somewhere else. So the groove which is there for binding to substrate, its design changes. If that design changes, then substrate will not be able to bind, and khalas. Okay, so inhibition that is there, as the last statement says, inhibition of enzyme can be lethal too, but then situation it is required depending on that. As I was explaining regarding it, we don't want the enzyme to run wild. That means churning out product after product. So hence inhibitors are there. So non-competitive inhibitor will do what? Will bind to somewhere else, and stop the process. Now, this end product inhibition that is there will be explained by a diagram over here. What it is showing is I am starting a process like a feedback mechanism, basically a feedback mechanism. Now, take for example, if the glucose level is very high, the diabetes condition that is, I know, like a blood glucose level is very high. Blood glucose is very high, a signal is sent, so body starts churning out insulin. So a cascade of reaction will occur. Since glucose level is very high, automatically a signal is sent to the pancreas and pancreatic cells that are there. They will start making insulin. The enzymatic process will be involved in making insulin. As insulin is released, it will start putting glucose level down. As the glucose level goes down, another signal is sent that we don't need that anymore. Glucose, the signal is sent that the insulin glucose level is within limits. No more insulin is required. So insulin production will stop. Otherwise, what will happen is if too much insulin is there, glucose level will go beyond the lower level and will lead to hypoglycemic condition and may lead to death. So this is what a chain reaction example is shown over here. First enzyme binds to the substrate, product is formed that binds to another enzyme, then the second one, the product then binds to the third one. Then the end product that is there formed after the third enzyme's activity binds to the first enzyme and alters its shape in such a way that a substrate is not binding. This is like a feedback mechanism. When this product is down, when this products that is the square product level is down, there won't be any one to bind. So then the green substrate will bind to the first enzyme, will be able to bind to it. And as soon as again the product is formed, end product is formed, more in concentration, it will bind to the first enzyme and again stop the process. So it's like a give and take going up and down, balance is maintained. Okay. So this is called as end product inhibition. The end product that is formed in this reaction, one after the other chain reaction, is stopping the process. But here the end product is not competing for the same site. It is not competing binding to the same site as the substrate, rather it is binding somewhere else and altering the shape of the binding site, which is prohibiting or inhibiting the binding of the substrate. As a result, no further reaction. Okay. Now, this graph represents the effect of competitive and non-competitive inhibitor. You can take a screenshot of this, giving a discussion, a brief difference between the competitive and non-competitive inhibitors. Previous slide. Okay, this one. Now, competitive inhibitor, as explained, this left image of the is self-explanatory. It's about the binding region. Now, the graph that is showing is when there is no inhibition, it is going at a very high speed. Then what is there? It is over here. It is not affecting. It is affecting the speed slightly. Okay, but not the binding pattern. So you can see the rate has reduced. Okay, let me see if I can explain this. See, this one is normal condition. This is normal condition. Here, what is happening now? This is at a very high. This is the rate. So it is touching the higher part and a very low substrate concentration, right? Now over here, what is happening that speed is reduced. You can see for this line, competition is there, speed is reduced. No, if competitive inhibition is there, its speed is reduced, not getting speed up, speed is prohibited, like velocity is reduced. And when non-competitive is there, you can say it's constant, that is a Michaelis-Menten constant shifts increasing. Now, in comparison to non-competitive, when competitive is there, what is happening is a substrate concentration. If the substrate concentration is increasing, the speed is shifting. You can see the shift from the normal process to the right. Okay. And in non-competitive, what is happening is substrate concentration is not having any effect on that as such. That is what it says. See over here, this line. In the case of competitive, substrate concentration is having an impact. But in the case of non-competitive, even though substrate concentration is increasing, but its effect is going down and down. So if you are comparing competitive and non-competitive from the graph, it is that competitive is affected by the substrate concentration, whereas non-competitive is not affected by the substrate concentration as such. Okay. Okay, quickly, answer is C. Okay, Shruti ji sahi hai. See, the question says what would be true regarding action of all enzyme inhibitors? They bind to the active site. Do they? Do all inhibitors bind to active site? That is not true. Only the competitive way. What about non-competitive? They are not binding to active site. So one is not correct. Cause a change in tertiary structure? Yes, they may cause a change in the structure. Then reduce the rate of enzyme catalyzed reaction? Yes. So the answer is C. Okay. Please read the question carefully. Next. What is the answer for this one? Anyone? Inhibitors of enzyme reactions form enzyme complexes. Okay. This statement is correct. Initial rate of reaction is decreased. Active site changes shape while forming the inhibitor complex. Inhibition can be this. So let's see. Is it 1, 2, 3, 4? Is it 2, 3, 4? Is it 1, 2, 3? C. When you're going for 1, 2, 3. Yay! So it is C. Good. Okay. So initial rate of reaction, well, degrees, active site changes, inhibition can be this. The maximum rate of reaction is increased, not true for all. Okay. In the case of competitive, yeah, in that case, the Vmax is increased. No, sorry, decreased. Fourth one that is there, fourth statement is not true. Yeah, all right. It's not applicable to all. Basically, it is talking about all enzyme inhibition. Okay, it is not applicable to all. The maximum rate of this, that means in this case, in the case of competitive inhibition, it will take time to reach there. So in a way, you can say the statement is false in this case. Okay, for this one,